Thank you for having me, and thank you all for attending this talk. So the field of 3D genomics studies the 3D folding organization of DNA inside of our cell nucleus. In each of our cells, we typically have about 2 meters or 6 feet of genomic DNA. And this length of DNA needs to be packaged and folded into the cell nucleus that's only about 10 micron in diameter. And this ratio of compaction proportionally is equivalent to packaging the circumference of Manhattan into a soccer ball. It's a tremendous task that our cell managed to do and do so in an orderly fashion. Correct 3D genome organization is essential for many genomic functions and for proper physiology and development. And altered 3D genome organization is associated with aging and diseases. For example, here you are looking at just coarse-grained DNA staining of a healthy growing cell, and you can see the DNA labeling inside of the 2 cell nuclei here, and the DNA staining inside of a cell that has gone through this process called cellular senescence or cellular aging. And just from this coarse-grained staining, you can see the distribution of DNA in the aged cell look drastically different. And other kinds of DNA folding changes are also associated with severe diseases such as cancers, progeria, which is an early aging disease, intellectual disabilities, etc. So how is our genome organized inside of cell nucleus? The overarching picture we have right now is organized in a hierarchical manner. So first, the double helical DNA wraps around the histones to form these nucleosome structures. And this complex of DNAs and histone is also called chromatin. And then at the other end of the spectrum, at the very large length scale, individual chromosomes, we have 46 of those. Individual chromosomes occupy distinct nuclear space when a cell is not dividing. And these structures are called chromosomal territories. So these are actually old knowledge. People knew these back in the 1980s. And in recent decades, thanks to new technology development, we now know much more about the intermediate length scale, how DNA is folded in between. And this detailed 3D genome organization is a major component of our current understanding of the 3D genome. And there are also other techniques that allows us to map the regions of the genome that are interacting with other nuclear components, such as nuclear laminar or nucleolus. And in today's talk, I'm mainly going to be focusing on some representative contemporary 3D genomics technologies that can map the 3D folding organization, this detailed folding organization in the intermediate length scale. So there are mainly 2 categories of contemporary 3D genomics technologies, the sequencing-based 3D genomics technique and the imaging-based. And let's focus on the sequencing-based first. And when talking about the sequencing-based technique, one must talk about the high-C technique or high-throughput chromosomal conformation capture developed by Professor Job Decker and team, because this technique really opened up the field of contemporary 3D genomics. So high-C aims to map pairwise chromatin contact throughout the whole genome. And this is how it does it. So first of all, the pieces of DNA that are making contact with each other naturally in our 3D genome are cross-linked through proteins that are coating the DNA, such as histones. And then these DNA pieces that are digested and cut by restriction enzymes. And these new end of the DNA are religated together by DNA ligase. And these novel junctions of this DNA are pulled down and sequenced. So through sequencing, one can map which piece of DNA was originally interacting with which piece of DNA. And this is done in a high throughput fashion throughout the whole genome. So here you are looking at a typical representative data from a high-C measurement, what we call a high-C contact map. So in this matrix, each row and each column corresponds to the genomic locus on chromosome 14. I think this is the long arm of human chromosome 14. And each matrix element, the color, indicates the contact frequency between the 2 genomic loci on the row and column. So a redder or the darker color means there's more contact probability. So in the same Hi-C experiment, we got not only the contact matrix of chromosome 14, but actually for all the chromosomes, and we have measurement of the interactions between chromosomes as well. So Hi-C is really a powerful technique, and in this first invention of Hi-C, the researchers actually did a population average measurement. So this chromosome 14 contact conformation is from averaging many cells and many pieces of chromosome 14 copy. After this first invention, there has been several lines of improvement of Hi-C, and one of the lines is the pursuit of single-cell and allele-specific resolution. Here, you are looking at the result of the deep-C technique, or diploid chromosome conformation capture, developed by Professor Sunny Siyuan team. And now this matrix is from a single cell. And you can see that in this single cell, for chromosome 1, actually there are 2 copies. There's a paternal copy here, and there's a maternal copy here. So there's 1 copy inherited from the father, and 1 copy inherited from the mother. And you can see these 2 submatrices showing the intrachromosomal interactions of each of these chromosome 1 allele. And you can see the interaction between them as well. And this is obviously in a female cell because you see 2 copies of chromosome X and no chromosome Y signal. So now with single cell contact information like this, one can actually fit a polymer model to best recapitulate the contact measurement. And this is essentially the reconstruction through polymer fitting the chromatin folding organization inside this very single cell. So one can infer the 3D folding information from the contact information. So both Hi-C and Hi-C-like technique, like deep-C, goes through this series of enzymatic reactions, as I just mentioned. The interacting DNA pieces needs to be first cut and then ligated. So what if 2 pieces of DNA are interacting in a very large complex, and after cutting, the arms of the DNA are too short to be ligated together? So there are these large complexes inside of our cell nucleus, such as nucleolus, right? And there could be DNA that are held together by these large structures. They are interacting over this long range, but using Hi-C, they won't be able to generate the Hi-C signal because DNA cannot reach each other. So can we address this issue? Professor Mitch Gutman and his team developed a SPRITE, or split pool recognition of interactions by tag extension. And in this technique, the pieces of DNA that are held together by the same protein complex, they no longer need to be ligated together anymore. Instead, through a series of split pool tagging reaction, the pieces of DNA that are interacting with the same protein complex are tagged with the same DNA sequence. And then the DNA pieces interacting through another complex would be tagged with a different tagging sequence. And then through sequencing, one can basically read both this tag sequence and the endogenous DNA sequence and map which pieces of DNA are interacting with each other by the fact that they share the same DNA tag that we added. So this technique is really no longer need the ligation reaction anymore, and it allows the mapping of very long-range interactions, such as those through the nucleolus and nuclear speckle. And it also allows the mapping of multi-way interaction, meaning that not just the pairwise interactions between 2 pieces of DNA, but if you have 3 pieces of DNA interacting together simultaneously, 4 pieces, 5 pieces interacting through the same complex, they are tagged with the same sequence, and they're going to be showing up as a signal, like they are all interacting together. So this multi-way interaction can be mapped as well. So very powerful technique. And then both SPRITE and Hi-C still requires this first cross-linking step, right? So the proteins that are on top of the DNA needs to be cross-linked together to form this complex in order to perform the later reactions. One can imagine that may still generate some bias, isn't it? Maybe some DNA regions will be less coated by protein than the others. Some proteins may be more fixable and cross-linkable than the others. Some very transient protein-protein interactions may gradually accumulate during the cross-linking reaction time. So is there a way to even avoid the cross-linking reaction? Professor Anna Pombal and her team developed genome architecture mapping, or GAM, that solved this issue. So in GAM, the 3D genome organization inside of single nuclei are flash frozen. They're frozen in the flash. And then these nuclei, they undergo this serial sectioning. So they're sectioned into thin pieces, thin slices. And the overarching idea underlying genome architecture mapping is that if two pieces of DNA, let's say the locus A and the locus C here, are interacting with each other, they're making contact very frequently, then very likely they will be included in the same thin slice. And if you just sequence these thin slices individually and sequence many of them, you can actually build a contact frequency matrix by looking at which pairs of genomic loci more frequently show up in the same thin slice, right? So the contact matrix can be built without the cross-linking reaction even. All right, so far we've talked about 4 sequencing-based 3D genomics techniques already: Hi-C, Deep-C, Sprite, and GAM. And you can see a common maybe limitation of these techniques is they measure contact, right? They measure pairwise or higher order contact, but they does not provide like this direct visualization of genome folding in 3D. They do not visualize the 3D folding path of chromatin directly. That's actually what the image-based 3D genomics technologies do and what my lab focuses on. So back in 2016, I led the development of a first-in-kind image-based 3D genomics technology called chromatin tracing. So this technique essentially sequentially labels, pinpoint, and distinguish numerous genomic regions along individual chromosomes of single cells by imaging and essentially super resolving this 3D folding path of chromatin just directly in situ. So this is based on a highly multiplexed version of DNA fluorescence in situ hybridization, or DNA FISH. In short, in the first step, we have this library of oligonucleotide probes, single-stranded DNA probes, that we hybridize to numerous genomic targets through just space pairing. And then each of these probes have a region that hybridizes to the genomic DNA. They also have an overhang region. And then in a series of secondary hybridization, we introduce these fluorescent dye-labeled secondary probes that binds to the overhang regions on the primary probes associated with one genomic locus at a time. And through this serial imaging procedure, we can then pinpoint and visualize with nanoscale accuracy the 3D positions, the X, Y, Z coordinate of each of these genomic loci that we label, and link the loci positions into the 3D folding path. So chromatin tracing really enabled the direct tracing of the 3D folding organization of DNA in single cells in situ. In this first invention of chromatin tracing, we traced 4 human chromosomes, and soon enough, chromatin tracing goes genome-wide. And this is a work led by Professor Xiaowei Zhuang and team, doing genome-wide chromatin tracing by DNA merfish, or DNA multiplex array robust fish. Without going into the details, I'm just gonna tell you that in this cell, for example, 1,000 genomic loci across the whole human genome are labeled, and they're labeled through this combinatorial barcode that's read out by this sequential highly multiplex FISH procedure to decode the barcode and also pinpoint their 3D position over 100 rounds of imaging. So really genome-wide chromatin tracing now. An advantage of chromatin tracing, besides the fact that it directly traces the 3D folding of DNA, is it's readily compatible with other modes of omic imaging. My lab actually introduced the first combined chromatin tracing together with multiplex RNA and protein imaging called MINA, the technique. And the latest development along this line is from Lang Kai and his team, Professor Lang Kai and his team, introducing 2-layer DNA-seq FISH plus. And in this technique, you can see they labeled over 100,000 different genomic loci together with 17,000 nascent RNA species, 1,000 mature RNA species, and 65 proteins. So really a highly multiplex technique now. And we talked about imaging-based 3D genomics and sequencing-based 3D genomics. And at the end, I want to mention something that's at the interface of the sequencing and the imaging-based technique. That's actually this technique called in situ genome sequencing. So in this technique, thousands of DNA tags are first randomly inserted into the cell nucleus, into the genome. And then these DNA tags, these randomly inserted DNA tags, are imaged and sequenced through this in situ sequencing procedure, to pinpoint their 3D position and also the tag sequence. Finally, the DNA from the cell is actually extracted and purified and went through this ex situ sequencing. That's the other sequencing-based technique used to sequence where are the tags inserted in the genome. So basically, they sequence the genomic DNA sequence next to the inserted DNA tag. And this way, one can basically map the endogenous genomic DNA sequences to their 3D coordinates inside of the cell nucleus, and essentially allowing chromatin tracing. So this is using part of the modality of the imaging and a part of the modality of the sequencing, really the unique technique. Finally, I want to highlight some of the complementary advantages of the different groups of techniques. So for sequencing-based technique, it's advantageous in terms of by default giving us genome-wide coverage, whereas the imaging-based chromatin tracing still requires people to pick which are the 1,000 or 100,000 genomic targets that you want to image. So it offers less genome-wide coverage. And the sequencing-based technique can also reach higher genomic resolution with finer cutting and deeper sequencing. For example, a micro-C technique that I didn't talk about today, another high-C-like technique, can actually reach single nucleosome resolution. And the chromatin tracing technique cannot reach that at this point. The imaging-based technique, however, gave us direct observation of the real 3D coordinate and 3D folding paths, as I mentioned. It also is quite compatible with other imaging modality, as I mentioned. And another hidden advantage is it by default gives you single cell and single allele information at much lower cost per cell. So the single cell version of the sequencing-based technique are still quite expensive. That's why currently most sequencing-based 3D genome study still use the population averaged version of the techniques. But the chromatin tracing is like much lower cost per cell and gives you this single cell information. And in part because of the lower cost, recently we were able to use chromatin tracing to build the first ever single cell cancer 3D genome atlas of any cancer using any technique. And we were also able to introduce this new technique called the perturb tracing that essentially combines chromatin tracing with high throughput genetic screening, allowing us to have hundreds of genetic perturbations in this single chromatin tracing study. And no high-C study, for example, can have hundreds of genetic perturbations in the same study, in part due to the prohibitive cost. So as a take-home message, I think the sequencing and imaging technique really have complementary advantages, and I envision in the near future they will coexist and collectively contribute to our understanding of the 3D genome. I'd like to thank my lab and also thank my funding sources, including NHGRI, thank TDCC, and also WashU in St. Louis for hosting this event, and thank you all for listening to this talk.
Thank you very much, Stephen. We have time for one or two questions. Potentially, please use the microphone. Barack?
Thanks, Stephen. I've had to go to a lot of gene expression talks in my life, and they all start with this famous picture from the Alberts textbook of DNA wrapped around a histone, those histones into 30 nanometer solenoids, those folded over into florets, and so on and so forth. I wonder, with all of these 3D genome techniques and the increasing resolution, has anyone ever seen the 30 nanometer structure or the florets, or is that something we should just sort of relegate to the textbooks and be done with?
Indeed, so a great question. So in recent years, people don't mention about the 30 nanometer fiber anymore, largely because I think the current consensus in the field is either it's not really existing in the native cell nucleus, or if it do exist, it's a very transient structure. Not only with the new technique, people don't convincingly see that, just using the old technique like EM, people now are saying that they don't routinely see this in the cell nucleus either. originally, people saw this, I think, from purified chromatin and they're quite artificial buffer conditions. So now people are questioning whether these are really physiological structures. Thank you.
Thank you, Stephen. That was very cool. I really didn't know so deeply about these 3D genome technologies, so it's good to have them all in the same place. I'm wondering, what is your favorite application of these techniques, like about an application that you can get, biological insights? What is the best application for it?
Great question. So this is going to sounds like self-advertising, but I am very proud of some of the recent work that my lab does. For example, in this cancer 3D genome iceberg, we had several new biological insights. First of all, we see the very intriguing 3D genome bottleneck in early cancer progression that we totally didn't expect. We're expecting, the diversity of 3D genome just should become monotonically increasing during cancer progression. But we see this bottleneck of 3D genome. 3D genome becomes more homogeneous in early cancer progression. We showed a new genomic function of 3D genome organization. We found through chromatin compartmentalization, the genes can be expressed in a more stable and more homogeneous manner. So previously, people say, this compartment, active compartment associated with active transcription, inactive compartment associated with inactive transcription, but the association is always weak. It's a weak significant correlation. So compartmentalization is not needed for gene activation. We found by compartment, the gene activation can become much more stable in comparison to the compartment-independent gene activation mechanism. We also showed new ways to use the 3D genome to call candidate cancer progression drivers. And we showed it works better than just using the gene expression information. So using the 3D genome, we can now better call cancer progression driving genes, and these are new therapeutic targets. So these are just some of the examples that we can derive biological and therapeutical insight from the 3D genome. Thank you.
Just have one question left.
I was curious if there's any known correlation between the expression rate of genes and its level of condensation. For example, genes that are always highly expressed, are they very accessible? And the other question is where does all the RNA hang out once it's made? Is that known at all?
So let me try to repeat the question to make sure I understand. Is the question about whether it is the case that when the chromatin becomes more accessible, the gene expression of the region will go up?
Yeah, of course, there's active unwinding before a gene is expressed, but I'm wondering, is there a more general correlation between condensation of a gene and whether it's expressed at high level or not?
Great question. Yes. Yeah. So the short answer is yes. There is a correlation between chromatin accessibility. So this is very fine scale compaction and decompaction of the chromatin, one can imagine, to gene expression. But this is one layer of control of the gene expression. So there are other mechanisms that can control expression. So in other words, if a region of chromatin is decompacted, it's not necessarily you will see super high expression from that region. There are other mechanisms controlling that as well. So there is an association, but it's not definitive, deterministic. And then the second question is about where are the RNAs made? RNA made? Is that right?
Yeah, before it's exported in the cytosol, are there any regions where, does the RNA stay associated with its DNA locus or?
Right. So the RNA, after it's made, it actually goes through the whole life cycle. And during this whole process, it interacts with many different components. Initially, it's made from the DNA locus. Indeed, the nascent RNA is associated with the DNA locus. Then it has to be translocated. If it's a protein coding RNA, it has to be translocated through the nuclear membrane and then interacting with ribosome to produce the protein. And finally, during stress condition, it may be stored in certain granule, and finally it will be degraded. Degradation can happen at different places as well. So it's a whole field, and it's a very exciting field.
All right, thank you.
Thank you.